Good morning, everyone. I'm going to wait just 1 or 2 more minutes for people to join, and then we'll get started. Happy Sunday morning. Good morning. Everybody can see me okay. Welcome to our 4th Sunday Symposium Big October presentation. I'm glad all of you could be here with us this morning. I'm really excited for today's presentation. Being diagnosed with progressive MS, one of the things that's kept me hopeful since being diagnosed almost 20 years ago is research, and knowing that every day there are hundreds or thousands of researchers around the world that are going to work every day trying to find treatments or a cure for MS. How can you not feel hopeful knowing that people have dedicated their lives to making our lives better? So in a little bit, I will welcome Dr. Tim Coetzee of the National MS Society, and he will give us an update on what happened last week at ECTRIMS 2021, which is the biggest conference of researchers. I wish I could tell you right now off the top of my head what ECTRIMS stood for, but I think I'll have to have Dr. Coetzee tell you, but it's the European Council of Treatment. There's MS in there. Anyway, this is the last week of Big October. Next Sunday is our closing ceremonies. We're going to give out some awards, have some of our top instructors pop in, maybe do a little bit of a Halloween seated dance party for those of you who want to join us, but we'll send out some information later about next Sunday. And one of the things I wanted to do before we got started today was make sure everybody had the chance to see my colleague and the other half of MS Hope, Sarah. So Sarah is going to spotlight herself for a second and unmute herself. This is Sarah. That's some feedback. Sarah is the one that's been answering so many of your questions, and I just want to thank Sarah for all the hard work. Big October would not be Big October without her, so thank you, Sarah, the face behind the emails. Okay, so the way today is going to work is like our past Sundays. If you have any questions that you feel like can't wait till the end, you can put them in the Q&A on the right. You can tap on that, and then I will kindly interrupt Dr. Coetzee while he's speaking if I think it makes sense to ask that question as he is going through his slides. If not, when we are done today, you guys can sign off, go right back to the homepage and click on the Q&A session, and then we'll get to meet with him and ask all our questions live. You can see him, he can see you, and he's very good at answering questions, as he has answered, I don't know, maybe 1 million questions for me over the years. And I think what I'll do is I will tell you all about what's coming up for Big October week 4 at the end of the session. So let's get started right away. I would like to introduce not only a friend, but really a mentor for me ever since I was diagnosed with MS. I've learned so much from him, and he has really motivated me to stay interested in research and really understanding how important it is that it is organized and directed. And as many of you know, all the money that MS Hope raises for research goes to the International Progressive MS Alliance, which Dr. Coetzee had a huge role in pulling together and working with people. The National MS Society of the United States really helps run that organization and bring everybody together. And I think that was one of my sticking points from the very beginning, that I used to ask him, like, who's got a list of every research project that's going on around the world? How do we know that 2 people aren't wasting their time doing the same thing? I know that, positive results are always published, but what about the negative results? Isn't it important that we know what didn't work as much as what did work? So I had all these questions I was always asking him. And now I think we're in a much better place 10 years later from when I started asking the questions to have those questions answered. So at this time, I would like to introduce my friend, my mentor, and one of the smartest guys about MS that I have ever met, maybe the smartest guy, Dr. Tim Coetzee.
Good morning, EJ, and thank you for that very lovely introduction. I'm honored. And good morning or afternoon, or I guess good very early morning to those of us those of you who might be joining us from the West Coast or possibly even Hawaii. As EJ said, I'm Tim Coetzee. I'm the National MS Society's Chief Advocacy Services and Science Officer. And so I have responsibility for those 3 areas of the organization's work, along with the International Progressive MS Alliance. I'm coming to you from my home office in Albany, New York, which, for those of you who may not be familiar with, New York is the capital of our state. It's about 150 miles, or not quite 250 kilometers north of New York City, if you're looking to orient yourself, if you're not familiar with where we are located. And really, it's a pleasure to come and join you today and give an exciting update, I hope, of the progress that we're making in MS research. And this comes from the ECTRIMS meeting, and I'll go through what that means, EJ, all those letters. And I'll be happy also to answer questions live while EJ poses them, as well as at the end of our time together. So why don't we get started? Let me pull up my slides, make sure that I can do that. I have some visuals that I'll share. And hopefully everyone can see it. Is it coming up, EJ? Perfect. All right. All right. So why don't we get started? So as EJ said, let's see if I can get this right. Okay, there we go. So I'm going to share a bit about some of the breaking news and some of the updates from the annual meeting of the European Committee for Treatment and Research in Multiple Sclerosis, otherwise known as ECTRIMS. ECTRIMS is one of the oldest and largest of the professional organizations focused on MS. It's, I think, been in existence almost 35 years now. And really brings together initially the European researchers focused on MS, but now it really has become the global annual meeting. When it last met in person in 2019, it brought together more than 12,000 researchers to spend 3 days just focused on MS research. And so this is the place where many people go, including those of us from the MS Society, to get up to speed on the latest research and also to see and connect with researchers from around the world. Just as a sort of other note, there are other organizations that have sort of a similar, what we call TRIMS acronym. There's the Americas Committee for Treatments and Research in MS. So that's a professional organization that brings together researchers in North America. So basically United States, Canada, but there's also similar organizations in South America as well as in Asia, really where researchers are all coming together to try to focus and make progress in their home countries as well as internationally. So this year, of course, because of COVID, The meeting happened virtually. So in theory, we were supposed to be meeting in Vienna, but that of course didn't happen. And so instead the organization pivoted and held a live virtual meeting that happened last week, in fact. While people couldn't get together in person, I think everyone, as many of us have done, have adapted to our current environment. And so 6,700 people, myself included, participated virtually over 2 days to hear again the latest updates. There were more than 1,500 presentations in various formats, either live, pre-recorded, as well as what we call poster sessions, where scientists organize their ideas into a visual representation that they then present and interact with people around. There's a lot of important information that came out, and I'll point you to some additional sources for updates at the end of my time together. And what I'm going to do in our conversation today is I'm going to share some updates, one related to COVID-19 and MS, and then go through a number of different areas that are what I'll just describe as Tim's curated sort of a selection of interesting topics and breaking news that are relevant to the MS Hope community. But please do note that is out of 6,700, out of 1,500 presentations, there's a lot there. So I was really trying to highlight some of the big things to attend to, but there's a lot that I won't be covering. So one of the big topics, as you might imagine, was the question about COVID-19 and MS. And obviously there's been a lot of interest in terms of what is happening in terms of how the disease affects people affected by MS. The big question about if a person is on a disease modifying therapy, how does getting vaccinated affect how does their DMT affect the vaccine and its effectiveness? And that has been, I'd say, the big overarching question, particularly for those individuals who might be on some of the stronger B-cell therapies or some of the other stronger therapies that affect the immune cells, like Gilenya. And so a research group in Israel has been tackling that question. And I suspect many of you who are watching today have seen quite a bit of the news flow about studies from Israel. And so Dr. Anat Achiran from the Tel Aviv University looked actually at 555 people living with MS, some of whom were not taking any disease-modifying therapy, some of whom were on one of the lymphocyte depleting types of therapies like Ocrevus, Rituxan, Gilenya, really to ask the question, for those individuals who are on a disease-modifying therapy, is their risk of a relapse higher because of the COVID vaccine? And also, did the DMT they were taking affect their body's ability to generate a vaccine against COVID? So what they found is that there weren't any increased risks for relapse after vaccinations. That's a really important point to make. The Pfizer BioNTech vaccine is safe in people with MS. And again, I want to stress, he only looked at the Pfizer BioNTech vaccine because that's the one that was available. I expect there's going to be more studies like this with Moderna and potentially also Janssen and the Johnson & Johnson vaccine and others. But this is related to the Pfizer vaccine. So he didn't find any increased risks of relapses. What he did find is that, for those individuals who were on a disease-modifying therapy, a stronger disease-modifying therapy like Ocrevus, Gilenya, and the like, that these individuals developed, their ability of their body to develop, a response against the vaccine and in effect develop the antibody response against the virus was not as strong as individuals who weren't taking a DMT, as well as compared to the normal population. And this is, an important finding, because what it's going to do, it's going to help us develop the guidelines and continue to look at, what's the advice we give to people with MS related to the COVID vaccine and their DMT and the timing for their DMT. I do want to stress, if you haven't gotten vaccinated, get vaccinated. The thinking about the timing of when you should take a DMT against vaccination is something to talk through with your doctor. I encourage you to seek, to visit the National MS Society's website. We have a lot of information there about getting the vaccine. But if you haven't already, I encourage individuals to get vaccinated. Don't let these findings stop you from going out and getting vaccinated. But the important take-home message also is that getting vaccinated does not put a person at any increased risk of a relapse, and that's also an important finding from these studies. There's going to be a lot more information around these. As I said, the society has done a lot of work in this area. We have an advisory group of some of the world's experts helping us develop guidelines for individuals living with MS around the timing of their vaccine with their DMTs. And so visit the society site if you want the latest information. And we also have some educational programs around that can also provide really breaking this down so that individuals can think about how to have a conversation with their doctor around this. So that's one of the big stories, COVID, lots of stuff happening there, lots of data being collected. But let's turn now to some of the science. And so I'm going to break this down in terms of 4 areas, which are sort of illustrated in this cartoon, where I've highlighted all the different cells, the immune cells, as well as the nerve cells that are involved in MS as we know it today. And basically, I'm going to cover 4 areas. What's happening in terms of advances as it relates to immune modification, targeting the immune system in MS. What's happening in terms of remyelination and neuroprotection, another big area to understand how we can actually improve and regain function. And then I'll touch a bit on symptom management and some work that's happening around improving quality of life, managing symptoms, and then wrap it up with a little bit of an overview of what's happening in progressive MS, because I know that's an area that I know in talking with EJ, it's something that through the Progressive MS Alliance we've been advancing, so I want to share some updates there. So before I jump into some of the advances in terms of treatments, I do want to illustrate, what we know within the cartoon about where all of our existing disease-modifying therapies are. And so I've put in those the purple and green boxes, all of the currently approved disease-modifying therapies for MS. I'm using the generic names, so I'm not using the brand names there, but those are the generic names for them. And one thing I'll draw your attention to is that a lot of these therapies, all of these therapies, in fact, are targeting the immune system. They're touching on what we call immune modification, with a real focus on what we call the T and B cells. The T and B cells are the major actors of our immune system that keep us healthy. But we also know that in MS, those cells are actually now malfunctioning, and the role of all of the currently approved disease-modifying therapies is to really change the nature of the T and B lymphocytes so that they don't cause the damage to the nervous system that happens in MS. And one of the exciting things that was reported at the ECTRIMS meeting was some advances and new treatments that are coming that start with the foundation we have with the existing disease-modifying therapies and take them further to get even more precise in terms of targeting specific types of cells in the nervous system that might be acting up and causing damage. And those are known as the BTK inhibitors. That's a shorthand for what we call Bruton's tyrosine kinase inhibitor. So that's the scientist in me has to come out every once in a while. The shorthand is BTK. This is an enzyme that's involved in different parts of how cells work. And one of the things that we have found is that the BTK pathway is very involved in the development of the B cells that are involved the B cells that are part of the immune system. And you might remember that for those of you that are on the disease-modifying therapy Ocrevus or Rituxan, that drug treatment specifically targets B cells and eliminates them so that they are not getting into the central nervous system and causing damage. And these are very highly effective treatments and have worked quite well. The BTK inhibitors take the work that has happened with Ocrevus and take it even a step further. And what they are is BTK are a treatment that is a pill that can get into the bloodstream, but also go to an individual's nervous system. So they get into the brain. And why is that good? The reason that's good is that what you want to do when you're targeting a B cell is you want to get the cells that are in the bloodstream, but there are also B cells that get into the nervous system as part of causing damage in MS. And if there were a way to get treatments into the brain in that way, you can take out the lymphocytes that are doing damage in both places. And so what's exciting about the BTK inhibitors is that they've gone now through phase 2 and now into phase 3 clinical trials. In phase 2 clinical trials, the BTK inhibitors have been shown to reduce immune activity. And so there's actually 3 of them known as evobrutinib, fenebrutinib, and talabrutinib. So three different molecules, 3 different companies. In relapsing MS, they have been shown to reduce relapses and immune activity, which is very exciting because, in phase 2 trials, if you can show reductions in relapses, odds are good that they'll show similar things in phase 3 clinical trials. The other exciting aspect about this work is that there are clinical trials being conducted in a couple of these, both in relapsing MS as well as in progressive MS. And so this is really an important development, because what it means for people with primary progressive MS is that there are going to be additional options, and additional options that are much more precise. And so the good news is we're going to have, potentially a treatment that can really increase its effectiveness and get at what's causing damage in the central nervous system. So I am excited about these developments, and really within the next couple of years, we're going to see, I'm hopeful, some additional treatments that are going to be joining the B cell therapies along with Ocrevus and Rituxan. So very positive. I'm super excited about that. But within that also, so that's the positive. We're also learning that there are some additional players that cause damage in the nervous system. And so those, I referenced these earlier. These are the red cells. We call them the microglia. Why are microglia important and interesting? The reason they're interesting is that these cells normally live in our brain and our nervous system and serve a very important purpose. And I will just call them, they're the street sweepers of the nervous system. The microglia, really serve a purpose of going around the brain, removing nerve cells when they die. If viruses happen to get into the brain, they serve as an early warning system and will clear out viruses. They play a vital role in maintaining the health of the brain. But unfortunately, in some situations when there's really a pronounced inflammation and damage to the brain, as happens in MS, the microglia can actually become hyperactive. And it would be a bit like if the street sweeper starts then, driving over people's yards and causing damage in other places. The microglia, in doing their work, then start damaging the nervous system. And one of the findings that was reported, an important finding that was reported at ECTRIMS, is that when the cells have gotten into that, negative mode, where they're, instead of being helpful and, cleaning out damage, are actually going in the other direction and causing damage, they are actually, damaging nerve cells and cutting nerve cells and killing them. And so this gives us a new target when it comes to treating MS that we can focus on. And the good news is that the BTK inhibitors that I mentioned before are actually targets for, can target microglia. And so there is the potential that the BTK inhibitors, in addition to eliminating the B cells that are causing damage in MS, will also target the microglia that are acting badly and cause them to actually basically settle down and no longer cause damage to the nervous system. So this is an exciting finding, both insights into what's happening inside MS, as well as what's happening in terms of the malfunctioning of a normal system in our brain. So continuing on, so that those two pieces were exciting developments related to COVID-19, some of the B cell inhibitors, some insights related to some of the newer cells that might be involved in causing damage in the nervous system. And so now I want to turn to another important update that we saw at ECTRIMS, and that's from a phase 2 clinical trial using a bone marrow stem cell therapy. And this comes from work by Brainstorm Cell Therapeutics, an Israeli company that is conducting important stem cell research in people with progressive MS. One of the things I want to point out is that the stem cell trial I'm going to be talking about here is a stem cell trial using mesenchymal stem cells. These are bone marrow cells that are treated and then engineered to create a proactive, positive environment in the nervous system. This type of stem cell therapy is different from autologous hematopoietic stem cell therapy, AHSCT, that you might be familiar with. I think there's been a bit of news lately, with the release of Selma Blair's documentary, where she documents her journey getting AHSCT treatment at Northwestern University. That's the more the bone marrow transplant type of therapy. That's a different type of therapy, and that's separate from what Brainstorm Cell Therapeutics is doing. The strategy that Brainstorm is using is that they take bone marrow stem cells from people with progressive MS. They then treat it to identify what we call the mesenchymal stem cells, and then they engineer those so that these cells actually become factories producing molecules that produce an environment in the nervous system conducive to promoting remyelination and neuroprotection. And so in this study, they took 18 people with primary progressive MS or secondary progressive MS, underwent this treatment. And the goal of this phase 2 clinical trial was to first show that this treatment is safe. That's always the first step, and to make sure that there is no adverse events associated with it. And then potentially also to see if there's any benefit. These individuals receive the treatment every 2 months, 3 doses every 2 months, by having it actually transplanted into the spinal canal that, where a person's spinal cord is. And what they found is that the treatment was indeed safe. And they also saw some indications that there were benefits for these individuals on measures of cognition, vision, and mobility. So, tantalizing signs of potential benefit. The society is involved in this work through Fast Forward, which MS Hope for a Cure has supported in the past. We're helping support their work to develop the tests to measure whether or not this treatment is showing effectiveness. And we're hopeful that the company will then take the next step of thinking about phase 3 clinical trials in progressive MS. And I want to stress these trials are only in progressive MS because the FDA has made very clear that these type of stem cell trials should only be conducted in primary progressive MS or secondary progressive MS patients. But exciting news on that front. Another area that I want to highlight is also myelin repair, which is something that I know EJ and I have talked about at length, a topic near and dear to our heart. And so, the upshot is that, from ECTRIMS is that we still have more work to do. There was reporting from a couple of clinical trials looking at different stem cell therapies, one called opicinumab and another one called elezanumab. Opicinumab coming from Biogen, elezanumab coming from AbbVie. The disappointing results were that while the treatments were safe, they did not show any benefit in terms of promoting myelin repair in the measures that those companies used for those trials. So a disappointment there. That we tested these and they didn't work. There was additional positive reporting about a different trial, though, of a drug called bexarotene. This is a drug that's been used for various other disease indications, and previously had been reported that this drug showed potential benefit in myelin repair. But at ECTRIMS, what they reported is that bexarotene, the individuals that went through that trial, actually showed improvement in the ability of their eyes and to be able to have a light shown in it, and then the brain registers whether or not the person saw that light. And why that's important is that it showed that in those individuals who were treated, the ability of the brain to process that information was quicker, which indicates that there was actually some myelin repair happening in the visual system, in the optic nerves of these individuals' eyes. So that's a promising indicator that there's at least, one drug out there that shows that, myelin repair can happen. Now, important caveat I have to say is that the bexarotene in this clinical trial also showed some unfortunate side effects, particularly related to an individual's thyroid. And so they're not going to take bexarotene forward, but they're starting to look at other molecules like bexarotene that may have myelin repair-like properties so that we can take those into clinical trials. So it's a little bit of a good news, bad news, but these are the kinds of work that we have to do to keep the field moving forward. Let me just stop here and ask EJ, how am I doing on time, EJ? And if there are any questions that have popped in while we're waiting.
You are doing fine on time. We had one question, and this is maybe the question that everybody has, and is it so slow? Why is it taking so long for progressive MS research to have any really outstanding progress?
Certainly. So if I could ask the questioner to hold their thought until the very end of my session, and I'll have some additional insights that perhaps can address that. So sit tight, and hopefully, if I haven't answered it at the end, we can continue on in the Q&A. So
I will ask one question about this slide since you asked. So obviously, both Biogen and AbbVie spent a lot of money on these trials, hoping and thinking that they would show positive results. Mm-hmm. Do we know why they didn't show positive results?
Yeah, so there's a couple. It's a little. I think they're still trying to sort out why they didn't show results. Likely, there's a couple things in play. And again, this is Tim's speculation. So just I'll own that. I think for both drugs, they were targeting different ways to try to create an environment that was more supportive to promoting myelin repair. And one of the things we've learned over the years of studying myelin repair is that in order for new myelin to be put around a nerve cell, you need to have an environment that's conducive to that. And what we're learning is that when nerve cells get damaged, you could think about it almost as though if you were looking at a wide open field and then there were fences that are getting in the way. In effect, that's what's happening as new myelin is trying to come forward. It hits this fence of leftover damaged material or different things. And both of these drugs, in terms of what had been found in animal studies, remove some of those barriers. And I suspect what we're finding is that there's other barriers that get in the way, in addition to the molecules that they were targeted, that there was probably no one sort of solution. So it's likely going to require a combination of different things, as well as potentially thinking about what's the right timing for a myelin repair therapy. Unlike our disease modifying therapies with individuals can take over time, my hunch is that there is a window of time that between when loss of myelin happens and then myelin can come back, and you need to figure out how to get in there and stimulate those stem cells that are present within our brain to lay down the myelin after the damage has happened. So my suspicion is that it's a combination of those factors, EJ. I know that's, again, I could probably imagine some of your listeners, our viewers saying, there's the scientist again, more research that needs to be done. And, I'll say I recognize that it's frustrating that we haven't quite figured out what are all those barriers that are getting in the way. The good news is I think we're identifying those barriers and we'll be able to lower those hurdles. So that's a positive.
All right, thanks.
So let's talk about some other aspects though that really excited me. So there's some other novel myelin repair strategies that were talked about at ECTRIMS. So one is a really promising and interesting, and you might think somewhat unusual idea and molecule. And so there's a new treatment called CNM-AU8. And you might be asking, well, what is CNM-AU8? Well, CNM-AU8 is a molecule developed by a company called Clene, C-L-E-N-E. And what this is these are actually gold nanocrystals. So really tiny, tiny crystals of gold sort of organized into this molecule that can actually be given to individuals. And what Clene has found is that this molecule increases the energy metabolism in the brain, and it increases the ability of the powerhouses of our brain, otherwise known as mitochondria. They're kind of like the turbines, the electrical turbines of our cells that gives us all of our energy. This molecule actually increases the energy metabolism, and why that is really important is that making myelin actually requires an enormous amount of energy. We sort of underappreciate that fact that myelin is something that it's a very energy intensive process. And what this company found is that in the clinical trial, using various MRI images, a person's the energy metabolism in an individual's brain was increased 10% over the clinical trial period. And that's a huge number. I mean, most of us lose about 0.5% of our energy metabolism each year. And so to be able to get back 10% is an enormous increase. And now the company is looking at taking the next step of actually, doing additional clinical trials to show whether or not, this can promote remyelination and protection of the brain. One thing to note is the society was also involved in this study, funding CLEAN through our Fast Forward program, again, to develop the measures they need to test these. And the last piece, some of you may have heard about, is a report from a company called Atara Therapeutics. They have a product called AT188. AT188 has a lot of interest because it targets T cells, targeting Epstein-Barr virus. But one of the interesting things that they have found in their trial, which was looking to see whether or not AT188 could in fact have benefit for people with MS, is that when they looked at the individuals using MRI, they saw signals of potential promotion of remyelination. So this is a bit of a surprise, needs to be replicated, but it goes to show you, I included it goes to show you how sometimes when a clinical trial is done to ask one question, when you sometimes can find other insights looking in other places. And so we're going to monitor this to see, but I'm hopeful that other strategies like these will overcome some of the barriers that I talked about in my last slide. So it's a bit of, again, good news, some promise, some challenging news, but I will also say the community is continuing to learn from the failed studies to be able to produce new and better treatments for remyelination. So I'm hopeful that when I come back, I'll be able to report additional insights moving forward. So then just to wrap up quickly, some other important pieces related to symptoms and quality of life. This is an area that's gotten a lot of focus, and there was a special session on symptoms and quality of life. And one of the fundamental things that came out of this is the importance of exercise. And Dr. Ulrich Dahlgest presented an important presentation where he looked at exercise and its benefit both in the early and later stages of MS. And what he has found is that exercise, it's safe, and it actually affects the symptoms across all stages of MS. So wherever someone is in their disease journey, the encouragement is to exercise and to really do the exercise that's feasible, and it shows benefits across the lifespan. And it also, in his findings, may have some disease modifying effects, which is also a positive. So for those of you who are watching, there's lots of videos on the MS Hope site around this. I would encourage you to visit those, participate in that. The society also has a lot of wellness and lifestyle-based resources. The fundamental thing is, doing activity really is an important aspect of managing MS, and I encourage individuals to continue to focus on that if you aren't already. There also was an interesting study that focused on digital tools and how they can assist with symptom management. And so in this case, Dr. Rainer Erling did a study where he looked at, could an app-based tool improve and really help people in terms of managing their spasticity. And what he found actually for individuals with spasticity is that after they'd gone through a period of training and then worked with the app over a 12-week period, that the app-based tool improved their spasticity and gave individuals agency to be able to make a difference in managing the spasticity through this interface. So it's an example of what a lot of the researchers in this area are looking at, which is to use digital platforms to be able to encourage individuals to both do exercise, to manage spasticity. There's also work being done around fatigue, managing depression. All of these digital tools are ones that are increasingly going to become part of our work. And I'd say watch this space. There's going to be a whole bunch more work in this area that is grounded in science that actually will also be about improving symptoms and quality of life. So a positive here. The app right now isn't available on the App Store, so don't go looking for it just yet. This was really an experimental platform, but I would say that in the not too distant future, we're going to see additional tools coming forward that people will be able to use. And then to try to answer the question about progressive MS, so I hope our viewer has stayed patiently with me. So there was an important presentation by Dr. Alan Thompson, who won the Charcot Award for his lifetime contributions to MS. And I've had the privilege of working with Dr. Thompson over the last 10 years on the area of progressive MS. And really, he was the founding scientific chair and leader for the International Progressive MS Alliance. I know EJ's met him, and really a significant contributor to understanding progressive MS. And this cartoon is something that he shared, which really paints the picture of how the progress we are making to understand progression. And what Dr. Thompson talked about is that what we're learning is, in the red box, you can see that there are all these what we call injury mechanisms, different things that cause damage and create, really damage the nervous system in different ways. The inflammation, the loss of nerve cells, the stresses that come from the damage associated with it, and that this accumulates over time. And our understanding is that it is the accumulation of these things coming over time that both causes the relapse we can see, that's what we call clinical expression, but also triggers what's happening in terms of progression of the disease. But I'll draw your attention to the two boxes in the middle. And this is what Professor Thompson, highlighted, that our field is really beginning to understand. And that's the sort of the tension and the seesaw effect of our body having the ability to repair itself. And that's quite true. Our brain has the ability to repair itself, and that early on in life that we repair ourselves much more aggressively. And in MS, we see that there is actually what we call reserve, an incredible ability of the body to withstand damage and repair itself, but that this then gets lost over time, and that this is amplified also as we get older. And so that the issue of how the body repairs itself and changes over time is affected by what we call biological aging, and that these two work together to then, be the part, contribute significantly to what's happening in progressive MS. And so what Professor Thompson highlighted is that this gives us actually new targets to focus on. It gives us, targets to think about how does, if we can tackle biological aging, can we actually slow down some of these processes? Can we prolong the ability of the body to repair itself through different types of treatments? And so these are exciting new opportunities that we have in terms of tackling progression. It's an important picture he painted about how we understand when progression begins and where we can intervene in it. And then to also, he shared this slide, which is another example of how the field has evolved. So when he and I started working together in 2012, there were just 4 treatments that were being evaluated for progressive MS. Now there's 24 that are in clinical trials today, a number of different types of strategies. And what I can tell you, these are the ones that are in clinical trial. There are also a bunch that are still in the laboratory making their way to clinical trials. And there's actually, almost 100 of those in development. And so while I appreciate that it seems that it's taken forever for us to get to this point, as Professor Thompson said, progressive MS has taken center stage, and we've made quite a bit of progress. We need to do more. I will tell you that. I agree completely. We need more than 24. I'd like to have 240. But the field is really focused and really dialed in on moving that forward. And so then just to quickly wrap up before we go to Q&A, there's been a lot of new treatments in clinical trials. We've got a lot more work to do in myelin repair, but there are promising results. The findings from Brainstorm are that stem cells could protect the nerve from damage, even maybe promote a little myelin repair. Exercise can make a huge difference, and we have a clearer picture of what's going on in progression. So a lot of movement. I'll also end by giving a shout out that if you want more, I've just given you a sort of a snapshot of what's of the highlights, Tim's curated list with the help of my colleagues at the MS Society. You can get more information on a lot of this and breaking news at the National MS Society's website, including if you're interested in clinical trials. There's a clinical trial finder on the Society site that if you're interested in some of these trials that are recruiting, you can go there. And also my good friend John Strum will be hosting my colleague Bruce Bebo, who's Executive Vice President of Research at the National MS Society, where Bruce will be sharing his curated list of the highlights of ECTRIMS this week on the Real Talk MS podcast. So go over to John's podcast there, which the Society is proud to host, be a partner with John on that, along with our MS Hope for a Cure. So thank you, everyone. I know we have Q&A. EJ, I'll hand it back to you.
Thank you, Tim. That was really awesome. I know that I wrote down maybe 10 questions as you were speaking. So I think I'm going to ask you to stop sharing your slides.
I'm trying to do that now, if you'll pardon me. Excuse me. There we go. That should be
There we
better.
go. Now I can see you much better. So I think what I will quickly do is I know we had a lot of people. Thank you to the MS Society for sending out another email telling everybody about Big October. And since you are our guest this Sunday, we had a lot of new people join us. So I'm going to just take 2 minutes so that people can show you where you can find Tim's slides on our site, and also where the table is for the Alliance if you want to find out more about the Alliance, as well as the MS Society's table is focusing on COVID as well. So I'm going to share my screen very quickly. And oops, sorry, I did not think I was going to be there. There we go. Okay, so this is the home screen, and I guess I will start here as well. Right now, after we say goodbye, we'll click on join session, and you will actually just post presentation, and that's where we'll be able to be live with Tim Coetzee and ask questions. To find Tim's table that has the slides up here where it says Symposium Presenters and Wellness Instructors, you just click there and you can slide down, scroll down, and there he is, and you can click here. And that will a little bit more about him, and then here are his slides if you would like to print those out. I'm also going to go back to the home screen, and for those of you who have not had the chance to visit the expo, this is where we have so much information. You simply click there, and then unfortunately it's a double click, and now you are at the expo. And here we have some fabulous information from 18 comprehensive MS care centers on different topics. And then if you scroll down, MS organizations. So over here is a table about the Alliance, and the National MS Society's table is here. Real Talk MS is here as well. They're going to have and I met Bruce when he first started working for the Society and interviewed him on some research. So he's another one of my favorite people and mentors in MS. So definitely check that out, as is John Strum. So yeah, so this is where you can find out more information about a lot of different things. So that's that. I'm going to go back to our home and encourage everybody to sign. This is where you'll sign in for the last week. For those of you who are just joining us, we have some wonderful live classes and Q&A this week. And I think for those of you who received our email, hopefully most of you did, with a list of all our live classes, but all of our featured sessions are here. And for those of you whose first time it is here, if you go up to all sessions, today is, what is today's date? Sunday, October 24th. You click on this arrow, you can click on Monday, and these are all the classes that are coming up. So you can add them to your schedule. And if a class is live, I know we have a great class Wednesday. A lot of you had asked to see this live mindfulness introduction with Q&A with Donna Smith. All her classes have been super early in the morning. So we're going to do a live class with her Wednesday at 7, just one to highlight. But all the ones that are live will tell you live, and that way you'll be able to actually talk to our instructors. So that is a quick look at how to find what's coming on this week. As I said, next Sunday is our, actually I haven't even listed, our closing session. I hope everybody will join us there. And I'm not even going to thank Tim right now because we're going to see him in 2 seconds. Please go back to the home screen and click on, oh, where did it go? Hang on, everybody. Here's the Symposium Actions presentation. All right, we're going to make sure if you go to the home screen and you don't see the Q&A, which we just all saw was there a minute ago. And I know that many of you have been with us know that we've had some technical problems with Cvents, the organization that is the company that's been hosting this. I'm a little bit embarrassed, but we're all friends now. So Sarah is right now making sure to see why that isn't showing up. I'm going to see if it's in our all sessions. Okay, hang on. Sarah thinks she may have found the error. Let's just make sure that works. Let's go back to home. Here it is, post-presentation open discussion, October 24th. Okay, click on this join session. It is back. Go back to the home screen. Tim, we sent you the link, right? Click there. We'll see everybody in 2 minutes Q&A. Thanks so much.
See you there, everyone.
Bye.